Our first speaker this evening is David Haussler. It's my great honor to introduce him. I have a list of wonderful accomplishments that I'll read to you and let you know about, but one of his most brilliant accomplishments in my mind is he's married to an incredible woman who's here tonight too. So good work, David. He's a distinguished professor of biomolecular engineering at UCSC's Baskin School of Engineering. He and his team produced the first complete draft of the human genome in the year 2000, an accomplishment of which we are all incredibly proud. He's a Howard Hughes Medical Institute investigator, a member of the National Academy of Sciences, and a leader in the UCSC Cancer Genomics Hub. He won numerous awards, including 2011 Weldon Memorial Prize given by the University of Oxford. He's a visionary, a pioneer, a rebel, and a brilliant researcher who is leading the 21st century war on cancer. And what's not to love about a guy who calls himself a computer geek? So, with that, I welcome to the stage David Haussler.
Thanks so much for that wonderful introduction, and it's a pleasure to be here to talk to you. In the 1 minute that it took me to walk on stage, another person died of cancer in the United States. In the last 50 years, we've managed to reduce mortality from heart disease by 70%, but mortality from cancer by only 10%. Next year, cancer will replace heart disease as the #1 killer in the United States. We couldn't have made progress 50 years ago like we can today on cancer because cancer is an incredibly complex disease. It's caused by mutations that occur in the DNA of individual cells in your body and direct them to grow in an uncontrolled way, creating a tumor. The problem is that there are thousands of different types of mutations that can occur anywhere in the 3 billion bases of DNA in each one of your cells, and they can even occur in combinations. The complexity of this disease is staggering. It's not really one disease, not 1,000 diseases. It might be 1 million different diseases. In a sense, every tumor is different. No human being can understand the full complexity of cancer, but we can attack it with computer-aided analysis. So the message I'm carrying to you tonight is that cancer is actually a digital disease, and it's the digital analysis of cancer that will revolutionize the way we treat it. Now, not all of the medical centers and the medical research establishment gets it at this point, but we're coming on strong. It was a great, great day in 1985 when then Chancellor Sensheimer came up with the audacious idea of sequencing the entire human genome, all 3 billion bases of it. At this point, people thought it was crazy, and there was a lot of argument. But in fact, that critical meeting in 1985 is what sparked the Human Genome Project. In the next 15 years, we developed an amazing new technology that was able to actually sequence those 3 billion bases, and we got our first glimpse of this DNA heritage that had been passed on from parent to offspring for eons. The DNA definition of our species. At that point, in 2000, the NIH got into a little bit of trouble, as did the rest of the international community, because there were too many data files and too much complexity in all of the DNA from this 3 billion bases. And there's a wonderful story that a graduate student from Santa Cruz named Jim Kent just stepped in and actually put all the pieces together for the first time. That first glimpse of our human genome was posted from Santa Cruz, this campus, on July 7th, 2000. Thank you. it was that moment that we really, as a species, got a first glimpse at our own genetic heritage. Since then, the website that Jim Kent and his team have created has become a standard. We have hundreds of thousands of biomedical scientists that use it every day from all over the world. More than 3 million hits on the web per week. This resource is really exceptional. It cost a lot. In particular, it cost an enormous amount to obtain the first genome sequence. The 20 laboratories around the world that sequenced the very first human genome spent $300 million to get that DNA script. Now, you can get your own genome sequenced for $4,000. Probably in a couple of years, you'll be able to get your genome sequenced for $1,000. This is an astounding improvement in technology. It was you and Ashley who commented that if the automobile industry had improved at that rate, a Ferrari would cost 40 cents today. We often think about technology as almost being simultaneously synonymous with computers. They're the concept of rapidly evolving technology. And in fact, if you look at DNA sequencing improving by a factor of 10,000 in 8 years and compare it to computer technology, which improved by less than 20-fold in that period, DNA sequencing technology is probably the most dramatically expanding disruptive technology that's ever been created. Now, the NIH, National Institutes of Health, ran into another problem recently. 2 years ago, it became clear that the amount of DNA sequencing data that was being generated by all the sequencing projects was overwhelming the infrastructure that was needed to understand it. At this time, I was one of 5 directors of the Cancer Genome Atlas Project, the world's leading project to sequence different tumors and understand the DNA changes in individual tumors. So again, UCSC stepped up to the plate, and we built a database for cancer genome sequences for the National Cancer Institute that houses all of the DNA from all of the major cancer genome sequencing projects of the United States. It's called Cancer Genome Hub. It's built to hold 5 petabytes of cancer data. It's hard to imagine what 5 petabytes is, but it's a 5 with 15 zeros after it. That's how much data we actually are going to put in this repository. And already, hundreds of researchers, several hundred researchers from all over the world are downloading data. We've actually served more than 8 petabytes of data combined to the cancer research community since we built CG Hub. It's important not only to collect the DNA data to understand to allow us to understand the different mutations that we see in the different tumors, but we need to develop more computer algorithms to integrate that data into a picture of what's going on in each of the individual tumors and to search all of that vast amount of information for the subtle statistical patterns that distinguish one type of tumor from another. In fact, every tumor is a complex collection of changes that have been occurring during the time that that tumor has developed. Some of them starting early, and some of them the latest parts of the mistakes that lead the cell down that path towards cancer. My colleague, Josh Stewart, has actually been tapped very recently to organize the Cancer Genome Atlas's first pan-cancer analysis. With our first 8,000 different tumors, we had a chance to look between different types of cancer, to compare breast cancer to lung cancer to ovarian cancer, and see if there were molecular signatures that were similar. These data have now had an enormous impact on the community, and you can read about them in Nature. There have been many news articles over the last few weeks. We're starting to understand the true complexity of cancer for the first time by getting these data. But 8,000 genomes is still not enough. We will need millions of genomes to actually understand the incredible complexity of cancer. Unfortunately, 1.6 million Americans are diagnosed by cancer every year. So those data could potentially be collected. Are they being collected? No, for the most part. Those data on these tumors that are being collected are being siloed off into individual repositories within individual medical centers and not made accessible to researchers outside of those medical centers. This is a huge problem because it's only with the statistical power of 1 million genomes that we are going to really understand the full complexity of cancer. My colleagues and I have undertaken the task of creating an international global alliance and asked the medical centers to join this. We announced this in June last summer, and we've had an enormous uptake. So there are more than 100 now major medical centers, including Sloan Kettering in New York and many other famous medical centers that have signed on to think about this problem of how can we share the genome data that is being collected by this extraordinary technology and make it available for massive computer analysis to really attack its true complexity. I'm very happy to see this progressing, but it's unclear who's going to play the role of coordinating these data at this point. And I'm here to tell you that Santa Cruz has a possibility of playing a very major role in this. Why? Well, why don't the big medical centers share data with each other? A lot of that is competition. But Santa Cruz is viewed as a fair broker. We're viewed as a trusted partner. Hell, we're the guys that brought you the genome. We're the guys that put it on the web for free for everybody to look at with no intellectual property restrictions or anything like that. We're Switzerland. And because of that, we might play a key role in integrating these data. And I cannot stress how important it is that we build this integrated database and we build the detailed computer programs that we need to analyze it. We need to call on the geeks right now. It's the geeks that are going to understand the true complexity of cancer. And without them, we will be lost. So I'm asking you to think about this, to call on patient advocacy groups that you're a part of to stand up and demand the data. We should have a massive campaign, donate your data. Make sure that your medical institute is actually obtaining the DNA data and is willing to share it with scientific research. If we don't do this, we will lose an enormous opportunity. So let's not lose another minute. Thank you.